If you're new to the concept of a cell cycle, or if you've not watched my previous video on cell cycle, I'll give you a quick recap. But if you know about the cell cycle already, you can skip to the phases of mitosis with the timestamp. The cell cycle can be divided into interphase, M phase, and cytokinesis. Now in interphase, there's G1, S, and G2. The first gap phase, the S phase where the entire genome of the cell is replicated, and a G2 or second gap phase. After G1, S, and G2, the cell will enter M phase. In M phase, it can either enter mitosis or meiosis. Then, after mitosis or meiosis has been completed, the cell will undergo cytokinesis. Cytokinesis is nothing but division of the cytoplasm. So, once that the cell undergoes cytokinesis, the daughter cells are formed. Now, coming to mitosis, in mitosis we have 4 phases: Prophase, Metaphase, Anaphase, and Telophase. In some sources, there's also the mention of a Prometaphase, which is between Prophase and Metaphase. Now, I'm going to tell you something that has helped me make sense of the cell cycle and mitosis and how a cell divides. So, I hope that this helps you as well. Now, for a cell to undergo division, this is a cell, it has a nucleus. Forget about the other organelles. We have a cytoplasm and nucleus. So for the cell to divide, the organelles will be copied, and there will be 2 copies. Each daughter cell will have 1 copy each. All that's fine. But what are the major events that need to occur for the cell to divide? First, the cytoplasm has to divide. Then the nucleus also has to divide. Now in the case of a cell cycle, first there is nuclear division. Okay, nuclear division is followed by cytokinesis or cytoplasmic division. Now our nuclear division is what is known as mitosis. Mitosis is nothing but the division of one nucleus into 2. Okay. Now let's think logically. The cell has one nucleus. Now this nucleus has to give rise to 2 more nuclei. Okay. So practically what do we need? The goal is to divide one nucleus into 2 nuclei. Now the amount of genetic material has to remain the same. This is a cell with 23 chromosomes. Then the daughter cells should also have 23 chromosomes, or it's not mitosis. Okay, so the amount of genetic material has to remain the same, and the ploidy has to be maintained. We've already discussed ploidy in Embryology Basics 1. Ploidy is nothing but the number of sets of chromosomes. So if these 2 things are not maintained, what is the point of nuclear division? And if the nucleus does divide even without these conditions, it's not going to remain the same. So to get similar daughter cells, you need to maintain the amount of genetic material and the ploidy. Okay, so how do you propose that this would happen? Just think, if we didn't know the phases of mitosis, what would the cell have to go through to maintain these 2 things? First, say this the cell that we're going to take as an example has 2 chromosomes. The daughter cells have 2 chromosomes each. They have to be similar. Like we just saw, the amount of material has to be the same. The ploidy has to be maintained. This is, say, haploid. Okay, one chromosome, this is chromosome 1, this is chromosome 2. So the ploidy has to be maintained and the amount of genetic material has to be the same. So we start with 2 chromosomes. When mitosis is finished, we have 4 chromosomes in total. So what does that mean? The amount of genetic material has to double. So when you're starting out with X amount of genetic material, you need twice the amount to divide it equally in between 2 daughter cells. Okay, first thing. The next is the chromosomes have to be separated into equal halves for the future daughter cells. Exactly. Now if we have these 2 chromosomes, you cannot just cut one half of this and one half of this and give it to each daughter cell. No, it has to be equal. They have to be equally segregated for the daughter cells so that they resemble the parent cell. So the chromosomes have to be separated into equal halves. Now these 2 steps are what we're going to see in cell cycle and in mitosis. Okay, I'll mention these 2 things as we see the phases of the mitosis. And please remember, this is not something that has been mentioned in any book or something. This is how I make sense of things in mitosis and cell cycle. So let's keep in mind, the amount of genetic material has to double and the chromosomes have to be separated into equal halves. Okay, the chromosomes have to be separated, right? We'll keep these 2 points in mind and we'll proceed with the phases of mitosis. First, we'll see some parts, important parts. Okay, this is the cell. Okay, this is the centrosome of the cell. This in light blue, this is the nuclear envelope. So obviously this is the nucleus, and this is the genetic material. Okay, it's in a decondensed form, so that's why it's like that. Now the cell, it has, first, as we discussed in cell cycle, the cell has to go through G1, S, G2, and then it will enter mitosis. Okay, the 4 phases of mitosis: prophase, metaphase, anaphase, telophase. G1 and G2, nothing significant happens with regards to the genetic material, so I'm skipping G1. Okay, gap phase, the cell grows, it copies organelles, it produces proteins, all of those things happen. In S phase, what happens? There is a duplication of the centrosome. If you don't know what a centrosome is made up of 2 centrioles which are placed at right angles to each other. You can refer to this in Embryology Basics 3 video. Okay, so the centrosome is duplicated and the DNA is replicated. S phase is the phase where the entire genome of the cell is going to get replicated. Now, do you remember the first point that we discussed a few minutes back, that the amount of genetic material has to be replicated because you're going to produce 2 cells from this? So the first condition has been satisfied. Okay, so DNA replication has taken place. So initially we had like this, 2 strands like this. Now we have a replicated DNA. After S phase, the cell enters G2 or second gap phase. Again, no significant change with regards to the genetic material. So the cell will enter mitosis after the second gap phase. Now coming to the first phase of mitosis, which is the prophase. Now in prophase, there is visible chromosome condensation. This phase will be visible through a light microscope. You can actually see the chromosome condensing to form these structures. Okay, you can see the sister chromatids, 2 identical sister chromatids joined at the centromere. For those of you who don't know, this is a centromere in purple, and each of these, this is 1 chromatid, this is 1 chromatid, and these are identical and they're joined at the same centromere, and these are called sister chromatids. So we have 2 identical sister chromatids joined at the centromere, and the chromosome length will decrease and thickness will increase. Of course, we saw it was like a strand, or it was in a very decondensed form. So the more and more it condenses, it's going to coil upon itself and it's going to make a shorter structure but thicker. So we have these chromatids. And the next thing that happens in prophase is mitotic spindle begins to form. Now, if you remember the 2 conditions that we began this video with, first was that the genetic material has to be doubled. Second was that the chromosomes have to be split in half so that they can be equally distributed to the daughter cells. Now, this second condition, first condition was already satisfied in S phase. S phase, genetic material duplicated. So now we have equal amount of information, that we have twice the amount of information so that we can split it into 2. Okay. Now, for the chromosomes to split into 2 equal halves, we need something to separate these. Okay, and that function is going to be carried out by the centrosomes and these microtubules. These microtubules are every individual strand is a microtubule. All of these together, a cluster of microtubules, is called a mitotic spindle. In case it's not very clear, if you're having difficulty understanding it, you can watch the previous video. I think it's Embryology Basics 3 on centrosomes, microtubules and centrioles. So microtubules are these protein structures, protein-based structures. They form something called centriole. Centrioles are these things. Okay, this is one centriole, this is one centriole, and these 2 centrioles together form a centrosome, and from them we get microtubules. Okay, that's the essence of it basically. So these microtubules will come, they'll come and attach to the chromosome at the centromere, and they will pull them apart. So these are going to help us achieve the second condition of dividing them into 2 equal halves. So we'll see that now. So the spindles start growing. Okay, so as the spindles grow, these centrosomes are pushing away from each other. They start pushing away from each other. They move to the opposite poles of the cell. So the final goal is for the centrosomes to move to the opposite poles of the cell. Okay, they are propelled by the lengthening microtubules between them. So these microtubules, they'll keep pushing, they keep moving to the opposite pole. So they finally end up moving to the opposite pole. Now, why is it important that they move to the opposite pole? These microtubules, as I said before, they are going to attach and pull these chromosomes apart. So for that, they have to be on opposite sides so that they can equally divide them into 2 halves. Okay. Next is the prometaphase. In prometaphase, the nuclear envelope will fall apart. Now, the goal was that the microtubules will have to attach to the chromosomes and they have to pull them into 2 halves. But if it has to attach to the chromosome, then it needs to invade the nuclear area, and it's already having a nuclear envelope. It cannot invade it. So the nuclear envelope has to break. It falls apart, and then the microtubules invade the nuclear area and bind to the chromosomes. Now where will they bind? Supposing this is our chromosome. Okay, these are sister chromatids, this is the centromere. Now on the centromere we have this protein based complex, two complexes. These complexes are called kinetochores. So these microtubules from the centrosome, they'll come and attach onto the kinetochores, okay, and these will drag these sister chromatids apart. So this was in prophase. The microtubules have attached to the kinetochores on the centromeres. The next is metaphase. Before I explain about metaphase, you need to know what a metaphase plate is. So this blue line that you see, this is the metaphase plate. As you can see, it's going through the middle of the cell. So the metaphase plate is basically an imaginary line which is equidistant from the two poles of the cell. Okay, it's an imaginary line and it's equidistant from these two poles. So by the time metaphase starts, the centrosomes have migrated to the opposite poles of the cell. Okay, the chromosomes are lined up at the metaphase plate. So this is the metaphase plate and the chromosomes have lined up along it. Then the anaphase. Now anaphase is the shortest stage of mitosis, and anaphase is where our second condition will be fulfilled, that the chromosomes have to be divided into two equal halves for the future daughter cells. So the microtubules have attached to the chromosomes from both sides. Okay, now in anaphase, these sister chromatids will be pulled apart by these microtubules. These microtubules will keep shortening and the sister chromatids will keep separating. So the sister chromatids break apart and two halves of the cell will have equal collection of chromosomes. So the second condition is also complete. Our genetic material was replicated in S phase, and now in anaphase there will be two equal halves of the genetic material, two equal collections of the chromosomes for the future daughter cells. Now we have two equal collections of genetic material, but that's not enough, right? It has to form back into a cell. So the nuclear envelope and the nuclei start to form. So the nuclear envelope begins to reappear and the daughter nuclei will be formed. And the DNA will start decondensing. If it's like this in the form of chromatids now, if it's visible, it will start decondensing into that previous form, however it was before. And after this, the cell will undergo cytokinesis. Meanwhile, there has been production of proteins and copy of organelles and everything. So now the next thing to undergo is cytokinesis. The cytoplasm has to divide. Now for cytokinesis, supposing this is the dividing cell, a furrow will appear like this. This is the furrow, and it will keep invading until it becomes two separate cells. Now there is something important about cytokinesis, that is there will be a ring of actin and myosin. Okay, actin and myosin. This is called an actin myosin contraction ring. Okay, this ring will pinch the cell off into two halves, and this ring will appear in a plane which is perpendicular to the plane in which microtubules were present. So our microtubules were like this, right? So this plane will be perpendicular to it, and it will divide the cell into two halves. So here we have daughter cells. We started out with this amount of genetic material, and we have the same amount of genetic material in the daughter cells as well. So what were our two conditions at the beginning? First was the amount of genetic material has to double. It doubled in S phase. Okay, the amount of chromosomes, this was in S phase. The amount of chromosomes have to be separated into equal halves for the future daughter cells. So that happened in anaphase, right? This was only nuclear division. After nuclear division, what else had to happen? Cytoplasmic division. So cytokinesis has also taken place. So now we have two daughter cells with the same amount of genetic information as the parent cell, and the same number of chromosomes, and the ploidy has also been maintained. So this is a summary of mitosis. So this was a cell. Initially it entered G1 and then went to S phase. In S phase, the centrosomes were duplicated and the DNA material was replicated. Then the cell went to G2. Then it entered prophase, which is the first phase of mitosis. In prophase, there was visible chromosomal condensation, and we could see the sister chromatids joined at the centromere. Okay, and this is visible through a light microscope. Then in prophase also, the microtubules started to appear from the centrosomes, and these microtubules push the centrosomes away from each other and they reach the opposite poles of the cell. After that, there was the prometaphase. In prometaphase, the nuclear envelope started to fall apart so that the microtubules can invade and hold on to the chromosomes. So after the prometaphase, there comes the metaphase. So our chromosomes are aligned along an imaginary line called the metaphase plate, which is equidistant from the two poles of the cell. And then the cell enters anaphase. Anaphase is where the chromatids are pulled apart away from each other so that they can form an equal collection of two halves of the chromosomes. And then telophase. In telophase, the nuclear envelope starts to reappear and two daughter nuclei will be formed. In cytokinesis, the cytoplasm has to divide. The DNA material has already started to decondense, and in cytokinesis, an actin-myosin contraction ring will appear here. Okay, this appears in a plane perpendicular to the plane of the microtubules. So the microtubules were oriented like this, and this is the plane of the actin-myosin ring. So it pinches the cell into two halves, and then we have two daughter cells. So this was all about mitosis, and we will be discussing the numbers involved in mitosis, like the number of chromosomes and the amount of genetic material that changes with every phase of mitosis and the cell cycle itself, in the next video.